So shall we continue? Now we welcome Antonio Neumann Portela from Helmholtz-Zentrum Dresden-Rossendorf and University of Grenada.
Yeah. Yeah, great. Okay. So, hello everyone. Um, Today I would like to show you some results that I obtained during my PhD thesis. First, I would like to show you the outline of this presentation. At first, the introduction, followed by the physiochemical mine water characterization and the microbial community of the both mine waters, and the microbial uranium-6 reduction experiment in the mine water. To sum up, some conclusion. Well, at first, after the Second World War, East Germany produced around 260,000 tons of uranium, and this mining activity ceased in 1981 with the accumulation of radionuclides and heavy metals in the environment. So flooding and chemical technologies were implemented as a remediation technology. But due to the high cost of these conventional remediation technologies, the bioremediation emerged as an efficient alternative to this or complement to these conventional remediation technologies. Thank you. Okay, the bioremediation of uranium could occur mainly by two different strategies. On the one hand, the biomineralization, and on the other hand, under oxy conditions, and on the other hand, the bioreduction under anoxic conditions, where bacteria cells couple the oxidation of the rhizome donor to CO2 with the reduction of uranium-6 to uranium-4. And in our work, our motivation is the biostimulation of the native uranium-reducing microbial community as a strategy for the in situ bioremediation of uranium-6. Well, I will start with the physiochemical characterization. First, we're working with two different uranium mines, Berola and Pula, and both of them show a different uranium concentration. This uranium concentration is an important starting point because most studies focus on the bioremediation of high contaminated environments. Biochemical species under environmental conditions, we calculate the possible uranium species in both mine water by the geochemical speciation called geochemist's workbench. In the Berola mine water, we predict highly mobile calcium uranyl carbonate species, as you can observe here, this red dot. And in the Pula mine water, we predict the possible formation of uraninite. By QT RLFS and parafact analysis, we identified two different species in the Berola mine water. First, the calcium uranyl carbonate species, as we predict before by the chemical speciation code, and uranyl carbonate. Both species are presented here, and the red and green line, and you can observe how the fluorescence band match with the reference. Well, I will continue now with the microbial community. The microbial community was characterized by the total DNA extraction and sequencing the 16S rRNA and the ETS1 region by Lumina Masek. In the left side, you can observe the bacterial community, and in the right side, the fungi community. Regarding the bacterial community, it's mainly characterized by anaerobic facultative bacteria. We identify sulfur oxidizing bacteria, nitrate reducing bacteria, for example, sulfuricourbum, which show a high relative abundance in both mine water, sulfurobum, which show a high relative abundance in the Pula mine water, the green area, and sulfuromonas, with a high relative abundance in the Berola mine water. We identify as well iron oxidizing bacteria, for example, the genus Gallionella or Sideroxidum, and with a low relative abundance, sulfate producing bacteria, as the Sulfobaca, and iron-reducing bacteria as an identified genus of the family Geobacteriaceae. I will continue with the fungi community, and it's mainly characterized by fungi involved in the wood decay process. Pula show a high relative abundance compared to the Berola mine water, where the genus Cadophora show a high relative abundance. Okay, so based on this result, we're going to start with the microbial uranium-6 reduction experiment. First, we did a screening for electron donors. And for the setup, we used the human amine water and the anoxic condition and 3 different electron donors. First, the glycerol, based on the literature, and vanillic acid and gluconic acid, because the wood, the mimes, the walls in the mines are covered by wood. And wood decay product such vanillic acid or gluconic acid has been identified before in the human amine water. So we choose them as an economic alternative to the other electron donors. The following slide shows the results, and you can observe how the using glycerol as a electron donor and vanillic acid, we observe a high decrease in the uranium concentration, but there is no decrease in the uranium concentration using gluconic acid. So, based on the limit concentration of uranium in drinking water and discard water, we choose the glycerol as the best electron donor to optimize the uranium-6 reduction experiments. Then we use the same setup, the amine water under anoxic condition and glycerol as electron donor. Here I would like to show you how the bottles looks at the beginning and at the end of the experiment with a black precipitate formation at the end of the experiment. This diagram shows the main parameters during the uranium-6 reduction experiments, and we observe a decrease in about 99% of uranium-6, 54% of uranium sulfate, and 95% of iron at the end of the experiment. So under experimental condition, we calculate by the chemical speciation code the possible species in the uranium reduction experiment, and using the redox potential and the pH values measured during the experiment. And uraninite formation is predicted after the 40 to 52 days, as you can observe here. But the result needs to be confirmed by spectroscopic techniques. So first, therefore, we sampled 3 different samples during the experiment based on the decrease of the uranium-6 concentration in the supernatant, which is 20, 60, and 90. And you can observe how an increase in the black line corresponds to the reference, and the green, red, and blue lines correspond with the different samples. And with 90% of uranium decrease in the supernatant, we observe a high dominance for the uranium-4. Then we use XRD to try to get more information about this for the black precipitate, but we did identify uraninite. We identified calcite as the main crystalline structure. Probably we didn't identify the uraninite due to the low concentration of uranium and the amorphous appearance. And when we have a look and analyze this sample by scanning electron microscopy, we observe this beautiful calcite structure. When we analyze this calcite structure by EDX, we identify uranium in on them. And thanks to the backscattering electron detector, we identify possible uraninite nanoparticles on the sulfate of the calcite structure. Okay, to get more information in more detail about this, the black precipitate, we use Hertz-Sanes and EXAF measurements. Using the same samples, the 3 different samples with a different uranium decrease in the supernatant, I would like to focus in the last one. Here, this result show the data recorded by Hertz-Sanes and compared to the different uranium reference. We have a reference for uranium 4, 5, and 6, and we can observe the black line is our sample, and how this band does this peak fit with the match with the uranium 4. So there is a dominance of uranium-4 at the end of the experiment, as we observed before. But there is a peak here that corresponds with the uranium-5. So we identify uranium-5 in our samples. But these results are summarized here in this slide, where thanks to the iterative transformation factor analysis of the Hertz-Sanes data calculated, we are able to calculate the dominant of the of the free oxidation state in all samples. I will continue focusing with the 90 uranium decrease in the supernatant, and the speciation is mainly dominated, you can observe here, by uranium-4, 60%, and uranium-5, the 30% of uranium-5, and no uranium-6 was identified, was measured. I will continue with the EXAF data. Here you will observe the initial evaluation of the uranium L3H K3A weighted EXAF spectra and the corresponding Fourier transform. We observe a decrease in the peak at 1.43 Absorb, related with the uranium-6, and an increase in the peak at 3.64 Armstrong, related with the uranium-4. But when we when we analyze this mixture spectra, and we were able to isolate by ITFA uraninite-like phase. So that means that we prove the uraninite formation at the end of the experiment. Thus, our sample is the black line, and the red line correspond to the reference. Well, to get an idea about the cellular location of the uranium and the crystallography properties of these uranium-reduced products, we use a high-resolution transmission electron microscopy, and we observed these bacteria cells and electrodense accumulation around the bacteria cell. Using the EDX, we see the dominance of sulfur and iron in the inner part, and the other part we observe a dominance of uranium and iron. Based on the EDX data, we were able to calculate the elemental distribution maps, and we confirmed the dominance of sulfur and iron in the inner area, and in the other area the dominance of this uranium. Okay, and thanks to the high resolution transmission electron microscopy, when we analyzed the accumulation, we were able to identify these nanoparticles, and measuring the lattice space of them, and compare this lattice spacing with the database, we confirm that these nanoparticles are uraninite nanoparticles. Well, to sum up, the conclusion of this work are the following one. Glycerol is an excellent electron donor for the biostimulation of the native microbes to reduce soluble uranium-6 in the mine water. High potential of the indigenous microbial community for soluble uranium-6 reduction. Glycerol as a biostimulant for the native bacterial community to carry out the reduction of uranium-6 to uranium-5, and 4, in contaminated water with low uranium concentration. The formation of biogenic uraninite as a product of uranium-6 reduction, and further research is needed on the speciation and stabilization of the identified uranium-5. So, to sum up, thank you very much for your attention, and please feel free to ask, or now or during the lunch break. Yeah, thank you.
Thank you very much for this very interesting presentation. Are there questions in the audience or online? None online? Laureline? Yes.
Thank you very much, Antonio. So it's more a chemical question by curiosity. I am surprised that you find a U5, of course, you too. So I wonder if you try to analyze the black precipitate 1 month later or 2 months later to see if U5 is already there. Yeah, sorry.
Yeah, thank you very much for your question, Laureline. Yeah, not after 2 or 3 months, but after 1 month, we analyze the sample by Hersanes. We are waiting to analyze the sample by them as well, but we identify that we measure as well the uranium-5, but the amount of uranium-5 increased. Yeah, so we could prove the stabilization of the uranium-5 in this case, and probably there is a deep disproportion of uranium 4 to uranium 5. So because the uraninite, for example, the uranium 4% decrease, so there is an increase in the uranium 6. So probably due to this oxidation, there is an increase in the uranium 5 after 1 month under oxy condition.
Does it mean that the uranium it formed is not stable? And can be reoxidized very easily?
Yeah, for sure. Uraninite under oxy condition, it's quite easy to reoxidize. Yeah. If you want to improve this as to reduce the reoxidation risk, maybe another electron donor, for example, glycerol phosphate. We have a orthophosphate uraninite form, something like that. Yeah. Thank you very much for your question.
Are there other questions? No? If there are no further questions, I would like to thank you again.
Thank you very much.
And thank again all the speakers. And I think we can close the session now.